Imagine a universe of life, not stars. A universe you can hold in your hand. Instead of distant galaxies, picture entire worlds of living cells, tissues and organs, all contained within a single, delicate chip. This is a universe where biology and technology merge, where the mysteries of life are miniaturized and made accessible. This isn't science fiction. It's happening now, in labs worldwide. Researchers are pushing the boundaries of what's possible, turning imagination into reality with every experiment. We're building miniature human organs, tiny, living models of ourselves, on chips no bigger than a memory stick. These chips are engineered with precision, using living human cells to recreate the essential functions of our organs. These aren't just clumps of cells, they're complex functional systems that mirror the intricacies of our bodies. Each chip is a living, breathing replica, capable of responding to drugs, toxins, and disease, just like the real thing. For centuries, we've relied on animal testing to cure disease, but animals aren't humans. Their biology leads us down dead ends. Countless lives and resources have been spent on experiments that don't always translate to human health. Now we have a new tool, organs on chips, offering a far more accurate reflection of human biology. These devices are revolutionizing how we study disease and develop treatments. Like the telescope and microscope before it, the organ on a chip is a new lens, one that lets us watch human biology in action. It's a window into the living processes that define us. We can see a lung breathe, a gut digest, a liver process, introducing medicines and observing their effects in real time. This level of detail was once unimaginable, but now it's at our fingertips. This isn't just an improvement, it's a transformation. The way we understand and treat disease is changing before our eyes. Let's explore how these chips are built, how they function, and why they're a monumental leap forward. The journey into this microcosm is just beginning. We're learning to model ourselves, creating tiny avatars of our biology. Each chip brings us closer to understanding the secrets of our own bodies. This could lead to a future free from disease, and from the ethical dilemmas of animal testing. Imagine a world where cures are discovered faster, safer, and more humanely. The cosmos within awaits. The next great frontier isn't out there among the stars. It's right here, on a chip in the palm of your hand. What is an organ on a chip? It's a marvel of microengineering and cell biology, representing a new frontier in how we study the human body. Picture a clear, flexible chip the size of a USB drive, etched with channels thinner than a hair. These tiny channels are carefully designed to mimic the intricate pathways found in our organs. These channels act like blood vessels, delivering nutrient-rich media to keep cells alive and healthy, just as blood nourishes tissues in our bodies. Inside, living human cells are arranged to recreate the structure and function of a specific organ. Scientists can use different types of cells to model organs like the lung, liver, or even the heart. For a lung chip, lung airway cells and blood vessel cells are separated by a porous membrane, mimicking the air-blood barrier. This allows researchers to study how oxygen and other substances move between air and blood. The cells come from human donors or stem cells, carrying our unique genetic code. This means each chip can be tailored to reflect real human diversity, or even model specific diseases. The chip isn't just a container, it's an active environment. Pumps and sensors can control and monitor the flow of nutrients, oxygen, and drugs, creating a dynamic system. We can stretch and relax the chip, simulating breathing or other physical forces. This mechanical stimulation helps the cells behave more like they would in a living organ. These cues tell the cells how to behave, just as they would in the body. The result is a more accurate and responsive model of human physiology. Without them, the cells lose their sense of direction and purpose. They become disorganized, and their behavior no longer reflects what happens in real organs. An organ on a chip is a living, three-dimensional cross-section of a human organ. It's a miniature, functional replica that brings the complexity of our tissues into the lab. It fuses biology and technology, giving us a window into our own bodies. This fusion allows scientists to observe, test, and learn in ways that were never possible before. It's not a whole organ, but the part that does the important work, brought into the light for us to study and understand. With organ-on-a-chip technology, we can explore human biology at a level of detail that was once unimaginable. How do these chips replicate our organs? It's all about recreating the right environment for living cells, but on a miniature scale. Scientists design these chips to mimic the physical and chemical conditions found inside our bodies, so that the cells behave just as they would in a real organ. The microfluidic channels inside the chip act like tiny roads and pipes, delivering nutrients and oxygen to the cells, and washing away waste, just like our own circulatory system does. These channels can be precisely controlled, allowing researchers to adjust flow rates and chemical concentrations, which is crucial for simulating different bodily conditions. In a gut-on-a-chip, for example, human intestinal cells line a flexible channel. Liquid flows through, simulating the passage of food, while the chip stretches and contracts rhythmically to mimic peristalsis, the wave-like muscle movements that push food through our digestive tract. This dynamic environment helps the cells function just like they do in the human body. Scientists can even introduce gut microbes into the chip, allowing them to study the complex interactions between our microbiome and our health. This helps us understand how bacteria influence digestion, immunity, and even how our bodies process medications. For a liver on a chip, human liver cells are arranged to break down drugs and toxins, just as they would in your body. By analyzing the fluid that exits the chip, researchers can see exactly how the liver reacts. Did it detoxify the drug, or did it create harmful byproducts? This level of detail is hard to achieve with traditional lab models. The chips are transparent, which means scientists can watch the cells in real time under a microscope. This allows for continuous observation of cell behavior, growth, and response to different treatments. Using fluorescent tags, we can track how cells respond to drugs, see them get injured, or even watch them recover and thrive. Time-lapse imaging reveals changes that happen over hours or days, giving us a window into the living biology of these tiny organs. No more guessing what happens in a mouse and hoping it applies to humans. With organ chips, we can observe human biology directly, using real human cells in a controlled environment. This direct observation is a game-changer for science and medicine, opening up new possibilities for research, drug development, and personalized healthcare. For decades, animal testing was the path to new medicines, but animals aren't tiny humans. Their genetics and metabolism are fundamentally different. Over 90% of drugs that work in animals fail in humans, wasting time and billions of dollars. Sometimes, promising drugs are discarded because they didn't work in animals. Animal models aren't just imprecise, they can be misleading. Organ-on-a-chip technology changes everything. These chips use human cells in human-like environments. When a liver chip shows toxicity, it's human liver cells being damaged. Studies show organ chips predict drug toxicity far better than animal tests, sometimes catching dangers animals missed. This means we can fail faster and cheaper, focusing resources on the most promising drugs. We're moving from blurry approximations to high-definition models of human response. Every revolution has its pioneers. In organ-on-a-chip technology, companies like Emulate and CN Bio are leading the way. They're building standardized, user-friendly systems that bring this technology to drug discovery worldwide. Emulate, spun out of Harvard, offers human emulation systems, lung chip, liver chip, intestine chip, integrated with fluid control and data analysis. CN Bio in the UK is known for advanced liver models, enabling long-term studies of disease and drug exposure. Pharmaceutical giants like Johnson Johnson and Pfizer are adopting these platforms, seeing the potential for better science and business. The pioneers have built the ships. Now the major explorers are setting sail. For new medical technology to change the world, it needs regulatory acceptance. For decades, animal testing was required for drug approval, creating a bottleneck. Now, the FDA is accepting data from organ-on-a-chip systems. The FDA Modernization Act 2.0 allows drug developers to use chip data instead of animal data. Years of validation have shown these chips are reliable and predictive of human responses. As more companies use organ chips, confidence grows, and adoption widens. The FDA's embrace of innovation is paving the way for faster, more accurate, and more humane drug testing. The human body is a symphony, not isolated organs. To truly understand a drug's effect, we need the whole picture. The next frontier connecting organ chips to create a body on a chip, a living, interconnected system. Imagine linking gut, liver, kidney, and heart chips. A drug travels through and we watch its journey and effects. This isn't a simulation, it's a living microcosm of human physiology. Only a body on a chip can reveal cross-organ toxicity and complex interactions. While still developing, these systems promise to revolutionize toxicology, pharmacology, and personalized medicine. Beyond science and ethics, there's a powerful economic case for organ-on-a-chip technology. Drug development is slow and costly, with most candidates failing late in the process. Organ chips provide accurate, human-relevant data early, helping companies fail faster and cheaper. This saves time, money, and lives. Promising drugs can reach patients years sooner. The technology also opens the door to personalized medicine, testing drugs on a patient's own cells to find the safest, most effective treatment. The benefits of getting the right drug to the right person the first time are immense. We stand at a crossroads. For centuries, animal testing was necessary for medical progress. But science moves forward, and organ-on-a-chip offers a new way to understand ourselves. We've seen the science, the pioneers, the regulators, and the industry embracing this change. The technology is here working and improving every day. Now we must ask, if we have a more accurate, faster, and effective human model, should we continue animal testing? This is a conversation for all of us, not just scientists, but society. Is it time to close this chapter and fully embrace human-relevant science? The choice, for the first time, is ours to make.